Alright, so you'll notice that the first half of the course we really focused on the structures of microbes, right? And we kind of repeated that a little bit with our unknowns when you guys stain them and look at them under the microscope. The second half, you'll notice that we're really talking more about function. What can they do, right? Especially we're looking at some of these results today. What did your organism do given these particular conditions? So this is not all that different from anatomy and physiology, right? Where you look at the different components and then you look at what those allow them to be able to do. So some structural things are like a capsule, right? Does it have that structural component? Where now we're starting to look into functionality. A lot of the questions that we ask in the next half deal with fermentation, right, of sugars. So, and glucose is a common sugar. In the past, we used to do the Methyl Red and Voges-Proskauer tests tonight and the Phenol Red, and look at citrate catalase and talk about NRO2. And especially with doing the inoculations, which we've already actually done this semester, that was a monster lab, right? So we're saving these two for next time, and this is actually going to be included in one of the later ones. But these are what we call differential tests, right? Differentiate the biochemistry of your organisms. So we already did the lab work for tonight. We did this last time when we inoculated this special media. So these are already in the refrigerator. They look great. You guys are going to get some great results from these. We ran the Methyl Red VP test, but again, we're saving the catalase for next time. I did the controls, right? Remember, we do this to make sure that we know what positive and negative results look like and that our reagents are working properly. And so everything went as expected with our controls, so then you guys continued on to your unknowns and recorded those results. So we divided our sample from our MRVP tube into 2 tubes, right, 1 ml each. And then we ran the VP test and we waited. Most of us for positive results, we had them within 10 to 20 minutes. And we did the Methyl Red, which is just a pH test, right? And we were able to read that immediately. Catalase we'll do next time. So, what's a redox reaction? Anyone know? It stands for reduction and oxidation. It involves the movement of electrons, right? All these chemical reactions, for the most part, we're dealing with electrons moving. So, if you're oxidized, you lose electrons, right? So, if a molecule gives up those electrons, it's said to be oxidized. But then, obviously, those electrons have got to go somewhere, right? So, someone else gets reduced. So, most oxidations are also in the same equation a redox if it's a complete reaction, right? Those electrons have got to go somewhere. So, they're commonly referred to as redox reactions. Reduction, oxidation reactions. A little saying that I learned when I was in chemistry was oil rig, and it's a acronym for oxidation is the loss of electrons, reduction is the gain of electrons. And that's easy to remember, especially living now in Louisiana, right? We deal with oil rigs all the time, right? So it's oil, right? O-I-L. Oxidation is loss. Rig, reduction is gain of electrons. So again, they don't disappear. They've got to go somewhere. So if somebody loses them, someone else picks them up, right? Someone else is reduced. So in biological systems, also protons tend to move. And in fact, whole hydrogen atoms will move. And what is a hydrogen atom? Anyone know what its parts are? How many electrons does it have? 1. How many protons? 1. It's the most simplest atom, right? 1 of each. So when it loses that electron, it's just a proton. So hydrogen ions are sometimes just referred to as protons. And those are what we measure when we measure pH, right? Is those free hydrogen ions that are positively charged. They're basically protons. Sometimes, you know, the whole hydrogen atom will move, right? Its electron and its proton will stay together. So here, this molecule is referred to as an electron carrier because it does just that. It tends to pick up electrons. Its abbreviation is NAD+. plus, right? It tends to be in the oxidized form. It's lost its electrons, right? So it's positively charged. It is, right, nicotinamide is this component of the molecule. That's where the N comes from. So again, it's an acronym in itself. Adenine is this nitrogenous base. Anyone know where you would find this nitrogenous base? Where you see adenine, a whole bunch of A's. DNA, right? This is actually a modified nucleotide, right? This is what you would find in DNA, except it's two. It's a phosphate and a sugar and an adenine. That's one nucleotide. Here's another nucleotide, right? You got your phosphate, your sugar, but instead of an adenine, it's the nicotinamide, right? And so the N stands for the nicotinamide, the A for the adenine, and the D stands for the dinucleotide that you're looking at, right? So that it actually stands for something. Just like we say DNA all the time, right? It actually stands for something, right? It stands for deoxyribonucleic acid, right? So it's made of nucleotides, phosphates, sugars, and different nitrogenous bases depending on the nucleotide. In this case, it's two of them linked together. But what this does is it allows it to pick up electrons. So it will actually pick up electrons and protons, a whole hydrogen atom you'll see right here. So then it becomes NADH, right? And then you usually, because it took the electron from a proton, from a hydrogen atom, you are left with a proton. Do you see that? Positively charged here. So, but now this guy itself is not charged, but he has an extra hydrogen here. Okay, do y'all see that? Okay, the important thing is he picked up electrons, but in biological systems, sometimes the whole hydrogen atom will go as well, right? Or in this case, it got split. So there are pathways in which we break down molecules to release these electrons and be able to utilize them to create energy, right? So glycolysis is, just as it says, the breakdown of glucose, right? And it's one of the most common pathways that we use to break down glucose. We break it down into we break it right in half into two molecules known as pyruvate, or sometimes also referred to as pyruvic acid. These are slightly different molecules, but at this level, a lot of people use them interchangeably, right? We're not going to get into the specific chemical differences in these guys, right? But know that they are different slightly. But in this case, pretty much the same. The next thing that happens to it is it goes through what's referred to as a gateway step before entering into the Krebs cycle. And so we'll add coenzyme A to the pyruvate, and we're going to give off a carbon dioxide in the process. We're going to break carbon dioxide off of the pyruvate. And so we're going to release carbon dioxide, and then you're left with an acetyl group, right? The pyruvate has been reduced by one carbon. And that binds to the acetyl-CoA. This helps bring it into the Krebs cycle. So through the process of the Krebs cycle, we're going to continue to break off carbon dioxides. And in this process, we're also going to take electrons from the acetyl-CoA, right? So the acetyl-CoA is recycled right in the beginning. And we're going to produce 4 molecules of carbon dioxide. So anyone know how many carbons are in glucose? In one glucose molecule? Well, there's 4 here, 4 carbons here, right? And 2 carbons here. 6. It's a 6-carbon sugar. So those carbons are all going to become carbon dioxide through this process. Throughout the way, as I said, we're going to pick up electrons from this breakdown of glucose, and those are going to travel to the electron transport chain. Where's the electron transport chain in our cells? Anyone know? It's a series of proteins, and they're embedded in a membrane. What membrane inside of our cells? Where does this happen in our cells? Where are the energy powerhouses inside of our cells? Mitochondria. Right. That's the key word there, huh? powerhouses. So what happens is that there are proteins embedded in a membrane. A membrane is a barrier, right? Guess what we're going to push across that membrane? Protons, right? And then we're going to only let them go through a little hole to get back to the other side. And when we squeeze them through that hole, it's almost like water, right? When you turn it over a turbine, right, for when they use it to generate energy, right? It spins the wheel, right? Old-fashioned millwork, right, was just right next to a river, just had the water turn the turbine. In this case, it's going to be protons, right? They're going to be flowing through this little hole. And they're going to drive the force that's going to allow us to generate ATP. Another modified nucleotide, right? Adenine triphosphate in this case. We're going to actually add 3 phosphates to that nucleotide. There's a lot of energy in those phosphate bonds. It takes a lot of energy to put them together, but when you break that apart, you're going to release a lot of energy. Well, again, remember the electrons have got to go somewhere. In this case, at the end of the electron transport chain, if you're talking about us, what is going to accept those electrons? Anyone know? What grabs onto those electrons at the end of the electron transport chain? Oxygen. That's why we need it. Somebody's going to grab those electrons. Oxygen does. And it forms metabolic water. Right? So right here, right, those electrons are going to form, adding, and some protons added to the oxygen is going to form water. Organisms that respire anaerobically, it means that they're not using oxygen. So they might be using something else like hydrogen sulfide and making, or hydrogen sulfate and making hydrogen sulfide. All right, again, the electrons have got to go somewhere. So ultimately, we're oxidizing glucose, right? We're taking electrons away from it. We're releasing that carbon in the form of carbon dioxide. The free energy is trapped into ATP, so we can use it later. Reduction, remember, always accompanies. So for aerobes, what's being reduced? The oxygen is going to accept those electrons. And it forms water. So this is a simplified form of the 6-carbon sugar. Each one of these points would be a carbon. Right? So 1, 2, 3, 4, 5. These are also called carbohydrates because they're basically carbon and water. For each molecule of carbon, you have a molecule of water. That's what sugars are. So in this process, we can actually generate some right at the substrate enzymatic level. We can make ATP. A little bit. 2 ATPs. The rest of the electrons are picked up by electron carriers like NADH that we looked at. Those are going to go to the electron transport chain. So here we have our 2 pyruvic acids, right? And then the pyruvic acids, again, we're going to get rid of a carbon dioxide. We're going to pick up some more electrons by the electron carrier. And then we have our acetyl-CoA, which can then enter into the Krebs cycle. See, everything's accounted for, right? So this is also sometimes referred to as the tricarboxylic acid pathway. And the reason why is the first molecule in the pathway that forms is citrate or citric acid. And if you can see, it has 3 carboxyl groups. So that's why it's tricarboxylic. It's also why some people call it the citric acid cycle. But it's actually named the Krebs cycle, and there's actually one more name, but you know, if you're the second name, you tend to get dropped off, right? And so I can't even remember the other guy's name. I might have it in here. So oxalate, which is a 4-carbon, combines with the 2-carbon acetyl-CoA from our gateway step, and we get a 6-carbon sugar back. So this is simplified, right? Doesn't show you all the steps, but you can see we have more substrate level phosphorylation. Sometimes it's GTP instead of ATP, but again, it's very similar molecule. It's carrying those phosphates, that extra energy. We got more electron carriers that are going to be generated. And another one we haven't mentioned yet, FAD, becomes FADH2. Notice all the carbon given off as carbon dioxide, and the oxalate is recycled, right? So that just keeps going through this cycle over and over and over again. That's why it's called a cycle. It keeps going cyclically. So these guys are reduced. They've grabbed electrons, right? They're going to travel to the electron transport chain. So one note, one of the things we're going to look at next week is a citrate test. This detects on whether your organism can take up citrate from the media. Not whether it can make citrate, whether it can take citrate from the media and utilize that. And it's an important distinction. It's also going to test to see if it can break down and metabolize ammonia. And so they're actually going to be going in reverse. They're going to take in citrate and break it down, right, into oxalate and acetate. So those electrons go to the electron transport chain, which is embedded in a membrane. For us, that's the membranes within the mitochondria. For bacteria, that's within their cellular membrane, right? So these proteins are embedded in the membrane, and they're going to shuttle electrons. As they do that, they're going to pump protons from one side to the other. For aerobes, such as ourselves, the electrons go to oxygen at the end, and we form water. So we shoot them across. For FADH, it only shoots 2 sets of protons across. So it generates a little bit less ATP than NADH does. So for us, right, that's happening in the inner membrane of the mitochondria. Protons are going across into that interstitial space. And then the protons have to come back through this port, this protein port, which is also an enzyme, that's going to add phosphates to ADP to form ATP. Right? And oxygen's going to pick up the electrons and form water. This is called oxidative phosphorylation because, again, it's electrons that are allowing us to add those phosphates. One of the problems we run into being aerobes or living in an oxygen environment is that these toxic forms of oxygen can form. So especially if you have an electron transport chain and there's oxygen present, the electrons can jump ship before the end of the transport and form superoxide radicals. The bad news with these is they're highly reactive forms of oxygen that combine with other molecules and cause them to not work properly. The other thing that can happen is it can form another reactive form of oxygen that's not quite as damaging as superoxide radicals, but can be destructive, is hydrogen peroxide. And so we're going to look and test for, we're going to use hydrogen peroxide next time to test to see if an organism has an enzyme to break down hydrogen peroxide, if it has catalase. So as aerobes, we have to have this protection. We have to have catalase to break down hydrogen peroxide.
So on the bonus, someone put us with the peroxide first?
Yeah. Yeah. So when we do this test, right, we're actually adding hydrogen peroxide, and if the organism has catalase, it will break it down into water and gaseous oxygen. So we'll actually see bubbles. So a lot of people used to, and they suggest that you don't use it anymore for disinfecting your skin. A lot of people use it as an antiseptic. You can use it to clean other things. They suggest you don't use it for wounds because it's just as destructive to your own cells as it is to the bacterial cells. And there's an old wives' tale, too, that said that if it doesn't bubble, it means your wound isn't infected. No, if it doesn't bubble, it probably means your hydrogen peroxide is old and you mostly just poured water on yourself. It should always, if you were to pour it on yourself, it should always, always bubble. Because we're aerobes, right? We have this. We have catalase. Your cells should be breaking this down and it should always bubble. If it doesn't, throw it out and get some new stuff. That's also why you don't want to swallow it, right? That reaction would happen inside your body. It would be really bad. It's also why it's kept in a dark bottle, because light can cause that reaction to happen as well, right? So they're trying to minimize it breaking down into water and gaseous oxygen. Another clue that it's probably been there for a long time is that if you open the top and it fizzes like it does when you open a soda can, that's because, again, the oxygen gas is being released that has been building up while it was closed. Right? The good news is it's cheap, right? Like 89 cents at Walgreens. Go buy you another bottle. But don't pour it on your skin anymore. Soap and water and triple antibiotic is what they suggest. But you can use it to clean your clothes and your countertops. You can use it for all kinds of things. You can use it as a mouthwash, but I wouldn't suggest it. It's nasty. But they put it in most of those things nowadays, right? All right. So this is the overall equation, right? We can take glucose. We also need some water in the beginning. We're going to form a whole bunch of electron carriers, NAD and FADs. We're going to need to collect those electrons. As you can see, they become reduced over here. We're going to need the beginnings and the phosphates to make the ATPs and GTPs. And then we're going to form water. And all the carbon's going to be given off in the form of carbon dioxide. So oxidation, the loss of electrons, right? And the gain of electrons by our coenzymes. And this is at the substrate level. So most of the ATP is actually these guys going to the electron transport chain. So 3 ATPs for each NADH and 2 ATPs for each FADH, right? So if we do the math here, right, we get 4 ATPs from these guys, right? 3 times 10, we get 30, right? So altogether 34. The yield can be theoretically as high as 38 when you add in these at the substrate level, right? So 34, 36, 38. In eukaryotes, however, we have to expend some to cross that membrane in the mitochondria because glycolysis actually happens outside the mitochondria, but the Krebs cycle and the electron transport chain happens inside. So we get a actual yield, right? So that's at the substrate level, and then the oxidative level is probably around 29 to 30, right, from cellular respiration. So we get quite a bit of energy from that, from one molecule of glucose. These are some cool videos, right, YouTube videos that show you guys the electrons moving, like that helps you understand and remember how all this works. So what if an organism lacks an electron transport chain? What is it going to do with all those electrons that the electron carrier picked up like NADH? No electron transport. So you got to give these electrons have got to go somewhere, right? We had a problem, don't we? So what do we do if we can't aerobically or anaerobically respire? What's the other metabolic option? Yeah, but then you're not going to get anywhere if you just store it, right? Turn it into something else. You're on the right path. So what's a different type of metabolism that we looked at even today? What did they do instead of aerobic respiration? Fermentation. Fermentation. The answer is fermentation. So what's happening in fermentation? Right? Yeah, well, no, that's one of the things, right? And there's lots of fermentation pathways out there. By no means are we memorizing any of these pathways, right? We want a general understanding of what's going on here. But there's a whole bunch of them listed in the appendices of your book. So there's going to give you guys a few examples. So say we have pyruvate, right? We broke down glucose into pyruvate, right? We've already got some electron carriers, right, from that. So one of the things that some will do is break it down further into formaldehyde. And in that process, they're getting rid of a carbon in the form of carbon dioxide. There's that electron carrier that has electrons from glycolysis, right? He's going to add it back to formaldehyde, and it actually forms one of the things we like. Ethanol and alcohol, right? Booze. So your good old Saccharomyces does this, right? Baker's yeast, right? Or brewer's yeast, right? This is what actually will convert sugars that you give it, right? And this is beneficial for producing alcohol. One of the things it'll also sometimes produce other than the alcohol is, what did we see in our tubes? What was the bubble? Gas. And so that's actually what helps your bread rise, right? Is the production of that gas that's going on. So another avenue it could take is, again, formaldehyde, but actually add one of the pyruvic acids to the formaldehyde and form acetolactate, and then they can remove a carbon dioxide and form acetonin. Does this look familiar to any of you guys from your homework?
Yes.
As an answer to one of your questions. One of the tests we performed today was testing to see if the organism produced acetonin, because it can later convert it into what? 2,3-butanol when it adds the electrons to it. So which test? Yes. The VP test, right? The first one we did, tested to see if your organism could do 2,3-butanol fermentation. What is butanol? What's ethanol? It's an alcohol, It's an alcohol. This is the only test that we did to see if your organism produces an alcohol due to fermentation. But we actually did not test for the presence of 2,3-butanol. We actually tested for the presence of acetonin, one of the intermediates in this pathway. This is different, ladies, from acetone. They used to take fingernail, or guys, fingernail polish remover, right? Acetone, different molecules. This is acetonin, right? So the VP test, Voges-Proskauer Test. So the acetonin, it accumulates in the media, and we actually were testing for the presence of that. So we'll go through the chemical reaction of that later.
So that question,
Mhm.
was present reactant as we have
Acetonin. Yep. So another thing that can happen is we can form lactic acid, right? This happens in our own bodies, right? When we don't have enough oxygen and they're still breaking down glucose for energy, we form lactic acid, which, as you know, can be damaging. But other organisms produce it and help us make things that we like buttermilk and cheese and yogurt, right? And you can see this is where they get their name, right? Lactobacillus, also that are used to make sauerkraut and sourdough and kimchi, right? All these sour things, it's because of the acid. Acids taste sour. And then lactobacilli are also important for the human vagina, right, to maintain balanced pH. Lactate dehydrogenase is an enzyme that's found in nearly all living cells, and because it's released upon death, this is when we can tell when tissues have been damaged. This is one of the tests they run to see if that enzyme is present in high levels in your blood, especially after someone's had a heart attack, right? They're looking for LDH levels, and that's why they're looking for it. Because, again, we want to be able to break down lactic acid, right? We don't want it building up, because it can go both ways. It can make lactic acid, but it also can break it down. A lot of enzymes can go both ways. So another pathway they could take is adding carbon dioxide and form oxalatate, right? Sounds familiar, right? Krebs cycle, right? Then they can add the electron carriers to it and produce succinate. Anyone know what type of molecule succinate is? It's an acid. It's an acid. And this is actually 3 steps of the Krebs cycle, so same enzymes, just going in the opposite direction, right? Just depends on what the cell needs, right? What's going to push things going in different directions. Krebs cycle doesn't always have to run in that way. Then they could even convert it further into propionate, another acid. Another pathway, right? Lots of other pathways. But the bottom line for fermentation is you're usually producing acids, alcohols, sometimes even ketones, and then also gases. Fermentation is the only one that produces hydrogen gas, right? But as we know, aerobic respiration produces carbon dioxide, right? But so can fermentation. So in your little tubes, we don't know if it's hydrogen, carbon dioxide, or a mixture of the both, right? Depends on your organism. And the pH of the media, right, when it was fermenting it. So we know it's one of these two possibilities. All right, lots of acids can be produced and some alcohols can be produced. So especially with the Phenol Red test, we were really looking to see how much acid was produced, right? We wanted to know if a large amount of acid was produced. And usually it's more than one acid. So the typical name for this test, other than the Methyl Red test, we're actually testing for what? Mixed acids. We're looking for a mixture of acids, right, with the Phenol Red test to see if it produced a whole bunch of acids. Now gases can be bad, especially hydrogen gas, right, which also can be, this can be done under anaerobic conditions, right? Fermentation doesn't need electron transport chain, right? So if you don't have any oxygen, you can ferment and you can produce hydrogen gas. And actually that's what commonly happens to animals when they're dead and they get that bloat. The most common pathogen that is doing that is Clostridium perfringens. This one's a spore former. It's found in the environment, so it can get into the intestines. It usually doesn't germinate, but when the animal dies, right, it becomes anaerobic, and then it starts breaking down the animal, and it produces the hydrogen gas and the carbon dioxide gas, which causes the bloat that will happen to the animals. Now, the Hindenburg is another famous example of also what can happen with hydrogen gas. It's explosive, right? And so it's lighter than air, right? But a spark can ignite it. And what do they put in the blimps now? Helium. Put helium. Airships of today. So some common products that you'll see is a lot of different acids, right? Formate, acetate, propionate, lactate, butanate, succinate, a bunch of different alcohols, ethanol, methanol, isopropyl, butanol and 2,3-butanol, some ketones, which are also acidic, like acetone, and then again the two gases. And remember, only hydrogen is unique to fermentation, but carbon dioxide can be produced by respiration, aerobic respiration. Interesting side note on methanol. Methanol is toxic, and it'll cause you to go blind. And the reason for that is that your optic nerve is the most active respiratory cells in your body. Like they're always working, right? And so they're always respiring, and so they can't when they're poisoned with methanol, and that's why you can't see. That's why you'll go blind. It'll damage the optic nerve. So make sure, right, if you're going to drink, you drink the right stuff. Yeah, not 200 proof either. So mixed acid fermentation, we already said. Which test is that? That's your Methyl Red test, right? What's the reagent used? Methyl Red. And what is it? It's just a pH indicator, right? Except we know that the pH for this one is 4.4 or lower. So what do these type of products, acids, do to the pH of the media? They lower it, right? Acids lower pH. So remember, protons are just usually from hydrogen atoms. Hydrogen atoms are just a proton and electron. Right, so when we're measuring pH, right, it's the partial pressure of hydrogen ions, right? So it's that hydrogen with it's lost its electron, it's just a proton. But these can create problems, right, in biological systems. So we measure this partial pressure of hydrogen ions, and when they're balanced, the pH is said to be 7. This is a inverse negative log of the concentration. So pH 4, right, that's a low pH, but that's actually a high hydrogen ion concentration, right? That's when something added to solution gives off hydrogen ions, protons. Basic solutions are when you have a lot less hydrogen ions. And so above 7, right, above neutral, and the pH scale goes from 0 to 14. Basic is also sometimes referred to as alkaline, right, conditions. Right, so high hydrogen ion, low pH. Low hydrogen ion, high pH. It's an inverse relationship. So there's lots of different pH indicators out there, and actually I was just reading up on some kitchen science, some of the fun stuff I'm doing with our middle school STEM program right now. And apparently red cabbage can be used as a pH indicator. It's kind of cool. So I'm going to play around with that next week. We'll see. But even crystal violet in different pHs, right? It'll change colors. Methyl red right there, as you can see, right? It's really in the acidic range of pH ranges. Here's phenol red. So notice it turns yellow at not quite an acidic pH. I mean, just barely acidic, right? So all those yellow tubes I handed out to you guys, those were just, could have just barely passed over the acid threshold, right? So this brings us to some of the questions you guys have in your unknown report, right? So when you do these comparisons here, right, for phenol red, this just means that, right, it's, I think it's 6.8. Yeah, that it goes yellow. So can a methyl red negative organism cause the pH of the methyl red media to drop below its initial value? What do you think? So if your organism was negative, which means it's not 4.4, does that mean it's still not acidic? It could be, right? It could be acidic. Actually, we should take out some. I might bring us some pH paper in next time. We'll measure the pH of y'all's methyl reds test media. Let's see. So the answer is yes. Does that make sense to you guys? Okay. The next questions might help you. Would you expect a methyl red positive organism to be phenol red positive? So methyl red is what? 4.4. So it's at 4.4 or lower. So therefore, if we looked at phenol red, should it be yellow? Yeah, look, way over here, right? If it's, yeah, anywhere 6.8 or below, your phenol red is also going to be yellow, which indicates it's acidic. Still with me? Next question says, will a phenol red positive organism, and what they mean by this is that it turned yellow, right? So, except for Tamara and Annie, everybody turned yellow, right? Everybody turned yellow. Was everybody methyl red positive? No. Right? And the reason for that is methyl red, that's a really low pH, right? Where for phenol red positive, it only had to be slightly acidic, right? Still with me? So the important thing that we're trying to show you here with these questions is that we're using two different pH indicators that have two different pH ranges, right? With the methyl red, we want to know, is it very acidic? Where the phenol red test, we want to just know if it produced some acid, right? Did it ferment a little bit? Even just a little bit. We'll go over it again. Will a phenol red organism necessarily be methyl red? No. Right? Not all you guys that were phenol red yellow, right? Were you methyl red red? Right there it tells you no. And again, that comes into that we're dealing with different pH ranges. Different pH indicators, different pH ranges. So notice Bromenthal blue is on here, I think. Yeah, right here. So notice that this one, we're actually going to be looking for it to turn blue, for the pH to actually go up in that media that we're looking at. Oh, this is Bromenthal. No, this is a different one. Although this one turns blue too. The one that we use next week isn't on here. It's this was Bromenphenol, but the other one is a different B, and another one that turns blue. So again, there's our methyl red, right? Oh, there it is, Bromenthal blue, this one. As I was like, that one can't be the right one, because I know that this one's around 7, it's green, but then when it gets above that, it turns blue. And when it's acidic, it's yellow. I played with that one, with I think this one with the kids today, or it could have been this one. I don't know. I have to look. Why do they have to have such similar names to do the same thing? Yeah. So, okay. The next one is, can your organism do 2,3-butanol fermentation? Which test is going to answer that for us? This is the Voges-Proskauer, the VP test. What reagents? Now, there's 2 for this, right? And you have to use them in the correct order. A and B. Barrett's reagent A and B. Reagent A is alpha-nethaphenol. I promise I won't make you spell it on a test. And Barrett's reagent B is potassium hydroxide, which is why we needed to be careful with this one and dispose of these properly, because that one's a strong base. What's the intermediate product that's produced in this type of fermentation that we're actually testing for? Acetonin. Not acetone, acetonin. Yeah. Okay. So what your organism can possibly do is take glucose that's in that media and break it down into pyruvate, right, acetylacetate, and then acetonin. And then it can further convert it into 2,3-butanol. So if the acetonin's present, when we add the alpha-nethaphenol, it binds to it. Then we add the potassium hydroxide, and this is why we got to shake our tubes, is that we add oxygen to this reaction by flicking our tubes vigorously and getting those bubbles in. It'll combine then with diacetyl, which is in the, this will form diacetyl, which will bind with the guanine, which is in the media. And this actually produces a molecule that produces a red color. But this is a chemical reaction, right? So this is why we had to wait, right? Because sometimes chemical reactions take time. Especially that enough of that product is produced that we can see it with the naked eye. So this amber color, that's not positive, right? Only red is positive. So for both the VP and the Voges-Proskauer test, red was positive, right? Or this one time we got these really weird pinkish colors, which I think Jessica, hers almost looked pink even, huh? Like a magenta. So for both tests, red is positive. So remember, it's just a pH test with the phenol red test, right? And at 4.4 or lower, it's going to stay red. In between, right, it might be kind of orangey. And then yellow above that. But this is still an acidic pH, right? And then we're actually forming this molecule that produces the red color for the BP test. So it's a chemical reaction. So there's just our results. You can see just a little bit, right? That's a weak positive there. So would a false negative result for the VP test more likely be attributed to poor sensitivity or poor specificity of this test? Yeah, it's a sensitivity issue, right? When you just don't see enough red color to see that it's a positive result. Maybe not enough acetonin, right? Not enough of that chemical forming for you to see. So there are other circumstances when the concentration may be low. For example, it lingers around for a long time, but not much acetonin was generated in the first place, right? So you have a really small amount in that tube that we tested for. Maybe your organism wasn't grown at optimum temperatures. For whatever reason, if it's below the limit of detection of the assay, you would get a negative result. Right? So for some organisms, of course, this would be a false negative if you actually were supposed to be positive. These are typically incubated for 48 hours. We've been incubating them for weeks, right? So hopefully, as you guys saw, we got some pretty quick, strong positives right away. Why were you told to shake the tubes after adding the second reagent? What did we need to add to this reaction? We needed to add oxygen. You've got to have that for this reaction to proceed too. So that's another thing that could goof it up if you don't do that. And then remember, the procedure for the phenol red is just a pH indicator, so we just added a few drops and we read those immediately. Red was positive and yellow. Anything not red, right, is negative. So orange would be questionable, right? So organisms like E. coli will produce, under certain conditions, when they're exposed to glucose, which is in that MRVP tube. They could have made it into acetonin, or they could also do this, produce a whole bunch of different acids like lactate, acetate, formate, and succinate. All those acids is what really brings down that pH a lot, right? So that's why we call these guys mixed acid fermenters. They produce a lot of acids. Then you'll also notice that this guy will produce hydrogen gas and carbon dioxide gas, as well as ethanol. Trying to remember if it specifies now. So why can we read the methyl red immediately? It's just a what? Just a pH test. Why do we got to wait for the Voges-Proskauer? What are we waiting for it to happen? A chemical reaction to happen, right? This can take time. Some microbiologists recommend re-incubating the organisms producing methyl red negative results for additional 2 to 3 days. Why would you do that? Why would you incubate it longer if it comes out negative? Let it grow, and if it grows more, it's going to produce more. It's going to produce more acid. So hopefully enough that it would get within that range. Again, we've already done this with your organisms, right? So if it was supposed to be positive, it came out positive for most of you guys. Alright, scientific method. And then that brings us to our phenol red tubes. But you guys had fun doing my new activity. I think it definitely probably helped. And the tubes were a whole lot better than some of these pictures. So this is the coded system that we use, right? A stands for what? Acid. And so this means what happened in this tube. It ate the sugar, and in this case for these tubes it's glucose again, and it produced acid. If you saw bubbles in the Durham tube, that means gas was produced, and we coded that with a G. This one, there was no gas, right? No bubbles, so we code that minus for the gas production. You notice I had a negative control for you guys, an uninoculated control, so you remember what the media looked like to begin with. It's kind of an orangey color. This one, it didn't change, right? So no acid, no gas production, so that was minus, minus. So why would that happen? Why would you not have any acid produced and no gas? What if you didn't put any live organisms in your tube? Right? So the other thing I usually tell my students is check for turbidity. Did something grow in that tube? Because usually, right, it's going to do one or the other. It's either going to produce acid, or in this case, it's going to be alkaline because it did what? It digested the protein. Right? And some organisms will do both. But they always pick something first. So as you can see, there's commercially available, you can buy the base broth, right, with no sugar added, and you can add whatever sugar you want. Or you can buy them with the sugar already with it in the durum tube. Lots of different sugar alcohol choices that you could test for. We looked at glucose. But we are going to look at lactose, right? And some of the other tests, and mannitol in the plate media that we look at. So we are going to learn about some other fermentation. So it has a pancreatic digestive casein. Anyone know where casein comes from? Milk, right? It's the protein that actually makes your milk white. That's why it's white. So this is a really good protein source. It's partially digested. Why would you want to do that? Why would we want to digest it for our bacteria ahead of time? Break up the molecules, make them smaller. Right, they're gonna eat quicker, right? Why do we grind up baby's food? So they can eat it, right, quicker. If you gave a baby a carrot, right, they have no teeth. They'd be sucking on that sucker all day, right? We want them to grow fast, right? So we basically give them baby food. Salt to balance it, right? In this case for ours, we have glucose. Phenol red, why is that in there? What is that? It's the pH indicator, right? So we can tell by just looking at the color of the tube what the pH is, and we don't have to do a pH test like we did with the methyl red. So here's our amino acids, here's our carbohydrate. This is the choices they have, right? Protein or sugar. Pick one. In general terms, what are they going to usually do if they're going to break down a sugar? If they're going to ferment pyruvate, what are they going to make in general? Acids, gases, and alcohols. Which two can we test for in this test? Can we tell if it produced gas? Yeah, that's what the durum tube's for. Can we tell if they produced alcohols? No. Can we tell if they produce acids, though? Yes, because the pH is going to change, right? Right? So which one of these can we not detect? The alcohols. So what's our pH indicator? Phenol red. Right? This one is going to turn yellow just below neutral pH, right? 6.8. So it's not much acid there. When it goes above 8.2, it's going to turn this cool magenta color. I don't know why he has negative here. Peter was unhappy with my pink. It wasn't pink enough for him. So here's our negative control, kind of orangey, right? What color does the media turn if acid is produced? Yellow, right? So here we see the yellow, right? We know that's acidic. So we code it A, but do we see a bubble in the tube? No, that's full of yellow media. So it's negative. What color does the media turn if alkaline products are generated? Right, this pink magenta color, right? So I updated it with a prettier, more pink picture. What does the germ tube help us detect? The gas, right? And again, we don't know if it's hydrogen or carbon dioxide, right? And it could be both. If glucose is fermented, what happens to the pH of the media? It drops, right? So it's going to turn yellow, right? So that's our yellow tube. We know fermentation happened, right? Because acids were produced because it's yellow. Peptone, which is a mixture of proteins, polypeptides in the media, right? This can be broken down, produce ammonia. What happens to the pH when that happens? It goes up, right? So then we end up with the magenta or pink tube, right? And we code this K, and this is for alkaline, right? We can't put A because then we'd have two A's, right? So we got to go with some other letter, right? So they went with K. So when given amino acids have a carboxyl and an amino group. They can be decarboxylated. You can remove carbon dioxide from them, one of the things they can do. Or they can deaminate them, remove the amino group. And that's when they form the ammonia, right? Is when they deaminate. And then they can, take electrons from that to get energy and feed that into the electron transport chain. So just like in our bodies, like if you don't have enough sugar, you'll break down proteins instead for energy. The problem with that is you can produce some acid products like ketones, right? And that could be bad. So some other tests we'll look at later, look if they break down particular amino acids, like this is orthine. They can break that down. Or phenylalanine is another amino acid that we can break down. And there are babies that are born without the ability to actually make or break down phenylalanine, and they can't eat it, right? And so they do a special test nowadays before you even leave the hospital to see if your baby has this deficiency.
So ketones can be produced from both fat and protein?
Yeah. That's why diabetics run into that problem, right? When they can't take glucose into their cells, they break down proteins and fats instead, and they produce ketone bodies, which are acidic. And that's why they can go into, yeah. So in our tubes, they have a glucose and protein choice. So what happens if they eat the glucose? They ferment it, don't they? So then they're going to produce acids, which are going to lower the pH, so you're going to see yellow. So you're going to know they ate the sugar, right? What happens if they choose the protein? They're going to deaminate it, right? They're going to produce ammonia. The pH is going to go up, which makes it's going to be pink, right? What happens if they're like me, right? They can't wait for the hamburger to be cooking. I kid you not, I do this all the time. Hamburger's on the grill, and what am I eating in the kitchen? A Snickers bar. Right? I'm getting my quick sugar fix because I can't wait anymore for the protein cooks. Right? So if they eat up all the sugar first, right, which some of your guys are like in the 48 hours even, right? They gobbled up all the sugar. It's gone. Right? So the pH has gone down, right? Your tube is what? Yellow, right? And then they start eating the protein. Yeah, it's actually going to turn what color first? Because it goes, orange is in between. And then if you let it keep going, you get those tubes that I gave you guys earlier, and it turned pink. But how did you know, other than the fact that I told you that it ate up all the sugar first, what else did you see in that tube? You saw the gas bubble, right? So that gas bubble would be a clue, right, as, oh, maybe it ate the sugar first, right? So for some of you guys, that's why we re-incubated them. Yours is going to turn pink, right? So you know yours eats up all the sugar, right? And then I'll move on to the protein. Now, if it produced gas, that's a good clue, right? But what if it was an organism that did produce gas? How are you going to know? This is why I refrigerate them, right? So that I could stop them before they ate it all up. Right? Oops. Thanks. All right, so this is where they look like, right? So there it is, right? Pink. So it went yellow. It's going to come back up, right? So here's some examples of these tubes. Were right at about 48 hours. The problem is when you shake these, right? This will kind of all turn yellow. But you see, this always looks like a cocktail to me. It makes me thirsty. But something like Sex on the Beach or something, right? But you see this one, you can see that gas bubble. So that's a really good clue, right? That it ate the sugar first, and then it's moved on to the protein. It ran out of sugar. But look at this one. There's no bubble, right? So some of you guys have ones like this too, that it's going to actually turn pink because we're going to let it go another couple of days. No, ate the sugar first, then moved on to the protein. They always choose sugar. It's fast energy, right? It's much more work to break down proteins. But some of them don't ferment at all, right? Yours went right to protein. Yeah. Protein first. Yeah. So this one would be coded acid, no gas, weak K, right? Where this one would be acid, gas, weak K, right? So there's actually some of you guys that think you're just this, but you're actually this, because it's going to turn pink. Right? So we'll look at those again. All right, so the interpretations of the tubes. So I forgot to tell you guys next time, I mostly just wanted you to fill in the code, and we would talk about this in class. So it's okay if you didn't fill in this table. You'll notice some of you guys were like, oh. I was like, it's okay. But here, in our tubes that we had, they had a choice, right? Sugar or protein. Okay? And these tubes, which we didn't do, were control tubes, where there's only protein in these tubes. So the only possible reliable result you could get in these tubes is either no change or that pink color that you get from breaking down protein. So if you get anything other than that in that column, then something went wrong, right, with your controls. And you wouldn't trust your unknowns, right? But we didn't do this. You guys understand? We didn't do standard base broth because we buy the already sugared stuff. We don't add sugar. So when you fill in your chart, remember AG means acid and gas, a yellow tube with a bubble. These are all possibilities that really can happen if you have sugar and protein, right? They could choose to eat the sugar and produce gas. But in these tubes, they only have protein. So how the heck did you end up with a yellow tube and a gas bubble? Is that possible? No, somebody screwed up, right? Somebody contaminated your tube with some sugar or something. I don't know, right? We would not trust this. Does everyone understand why we would not trust that? Right? This is a protein-only tube. How the heck did we get fermentation of a sugar? Somebody goofed. Now for the next one, could they, given protein, do nothing? Yeah, Tamara and Annie's ones would do that, right? They would be like, pfft. Or no, everyone else's but Tamara and Annie would be like, I give up. No sugar. Yours would ferment. You guys are the next one, right? Or actually yours are later on. But given the choice here between sugar, so this would actually be Adam's organism, right? Given the choice, it ate the sugar, fermented it, produced gas, and then when you gave it protein only, he's like, okay, fine, I'll eat the protein. Fine, I'll eat it, right? This is possible. Can you get sugar fermentation with gas in a sugar protein tube, and then all of a sudden in a protein only tube get acid produced? Where's that acid coming from? And why did you get gas the first time and you don't get gas the second time? That doesn't make sense. Right there, I would be like, oh, why didn't I get the same result, right? Not to mention, this is a protein-only tube. You should not see any acid from fermentation. So this would not be a reliable result. right? Given the choice, right? So this would be Annie and Tamara's, right? Given the choice between protein and sugar, hers chose protein. Turned pink. So of course it makes sense when we gave it just protein. He's like, okay, I'll eat it again. Right? No problem. Yum. Would you trust this if it would choose protein over sugar here? And then when given protein only, he didn't do anything? Right? So this is one of those ones where, right, did it even grow? Right? Did you just inoculate it with dead bacteria and that's why nothing happened? Could be, right? You wouldn't trust this though, right? You'd repeat. If given the choice between sugar and protein, right? Here it chose sugar, but it didn't produce any gas. Then when we gave it protein only, it was like, okay, fine, I'll eat it. Is that a reliable result? Yeah. Here, acid, no gas from sugar, right? Chose the sugar over the protein. Here we gave it protein only, but yet we're seeing acid and gas. Is this possible? No, shouldn't be. Somebody goofed. So, suppose you inoculate a slow-growing fermenter. After 48 hours, the tube, if it's minus, what does it look like? What color is it? Or orangey, right? No gas. But I already told you it's slow. You got to give it time. 48 hours is enough, right? So this would be a false positive or false negative. It's a false negative, right? Because I already told you he's a fermenter, right? Eventually it's hopefully going to at least turn yellow. Whether we get gas or not, right, is an issue. Can happen or not happen. So is this poor specificity or poor sensitivity of our test? Right, this is wow, you guys got this down, huh? She's like, oh yeah, I got sensitivity, right? Because we've got to get enough acid in there, right? So if he's slow about it, right, we're not getting enough acid, right? We're not getting enough color change to see it. Early formulations of this didn't have enough sugar in it. So 2 days later you came back and your tube was pink. So you think, oh, it didn't ferment the sugar. No, it did. It just ate all the sugar and moved on to the protein, right? So how could we fix this? Well, we still incubate for 48 hours, so what do you think they did? They put more sugar. Or we could, what? Instead of adding more sugar, instead of incubating for 48 hours, do it only for 24, right? Less time. Yeah. Peek in on them sooner. See what they've done. So I kind of fibbed about the early, huh? But that's okay.